Yes, thanks. Yeah, it's my pleasure to introduce to you the next speaker, Laura Mascotti. Laura did a PhD in Argentina. That's also where she grew up and got her education. She did a PhD at the University of San Luis, where she started to work on bioavailable monoxygenases, and that's also the reason that we met at some point. After finishing the PhD, she did do a postdoc, not only in Argentina, but at many places, I would say. Again, also in my lab for some time, but I think what was crucial for her next direction of research was her stay at a group of Janet Thornton. So she dived into bioinformatics. And more recently, she also has been working in a group of Joe Thornton. It's not family as far as I know, but he's a specialist on ancestral sequence reconstruction. So that's her direction in the recent years. And I'm happy that she joined my group a couple of weeks ago. She managed to fly from Argentina to Groningen. And well, she will tell you a bit more about what she has been doing on this topic and a little bit on what the plans are. Laura, floor is yours.
Thank you. Well, thanks for the introduction and for the invitation to these talks. So I'm going to talk about ancestral sequence reconstruction applied to cofactor-dependent enzymes. And basically, why is not? Okay. So basically, the fundamental question that I've been pursuing in my work during the last years is, how do enzymes work? And this is a question that you can tackle in very different angles. So the idea is basically to focus on which are the determinants of enzyme function. By the determinants, we mean which are the subset of the specific residues that accommodated into a certain three-dimensional form are able or give the enzyme the property of its own function. Yes, so, and specifically, we are focused on cofactor-dependent enzymes, mainly those using flavins, different kind of flavins or nicotinamides. And why is that? Well, because of two reasons. We come from biocatalysis and we are interested in biocatalysis, and of course, cofactor-dependent enzymes have a key role in different processes, but also because in the last time I've been working in the origin of life field, and actually cofactor-dependent enzymes seem to have a very important role in defining basically which was the metabolism of the first organisms on Earth. So how do we approach these questions? So basically we do it including evolution, but basically evolutionary biology. And let me show you an example, just a toy example to go through that. So usually in the lab, when we perform biocatalysis, we observe functional diversity among homologous proteins. For example, if we have one enzyme from a bacteria, we call it parlog1, it's able to perform the oxidation of a ketone to the ester. But another enzyme very similar from the same strain, a homolog, it's able to perform the sulfoxidation of sulfides. So the question is, how these two different enzymes perform different activities. So that is, which are the sequence determinants responsible of the observed differences that we are seeing. And our classic approach in general in biochemistry is the horizontal approach. That means to compare the two enzymes themselves. So it could be their 3D structure or their sequences, and try to find what is different among them, and then to design mutants. And of course, how to design mutants is a whole, a complete big field, and you can take very different paths to follow that, but basically, in the end, you will end up generating and testing different mutants. The thing is that to answer our question, maybe we will try to, we will have to test hundreds of variants of mutants, and many of them can be dead enzymes, usually they are, even though some of these could be less active or with unclear substrate preferences. So in the end, it's very hard to respond our question on which are the residues responsible of the different functions by this approach. So the idea to incorporate evolution is actually to include the history of the enzymes, and that is called the vertical approach. So basically, we know that extant enzymes, our two paralogs that from the example, come from a common ancestor because they are homologs. So they have gone through changes over time, which is basically evolution. So if we are able to somehow recover and characterize the sequences of the common ancestors among them, we might realize, for example, that the function of this modern protein actually is the same than the ancestral function. And actually that paralog 2 has a derived function. So all the changes between the two sequences do not strictly are responsible of the changes in functionality. For example, in this case, the switch in the function occurred in just this branch among these two ancestors. So basically two mutations are the responsible of the changes. This, therefore, dramatically drops down the amount of variance that we actually need to test to solve questions as how do we switch from one function to another? So this is the basics and the fundamentals of ancestral sequence reconstruction. So this approach allows us to recreate the proteins in the lab, basically to resurrect them, and actually to study the pathway that we inferred in the evolution directly. So test all the mutations in a certain branch. And it's important to clarify that the aim of the approach is to recover the phenotype of the ancestral protein. So what was the activity of the protein rather than its precise sequence. And this is because the approach is very robust towards uncertainty. But we can talk about it later. So in this context, in the last years, we have been working with different protein families. For example, here we perform ASR on the F420 dehydrogenases, which are enzymes very attractive from the biocatalytic point of view. And we were able to understand how they actually have developed a very specific substrate specificity for glucose 6-phosphate, which are the residues responsible for that. Also, on a different study, we worked on characterizing some ancestral fungal decolorizing peroxidases, and actually we were able to solve the structure and to understand how is the environment around the cofactor that modulates the activity of these enzymes. But today, I want to talk a bit more in detail of a third protein family that we have been devoted to studying the last years, that are the flavin-containing monoxygenases. So these are very popular enzymes, and I think one of the reasons is because we humans have 5 different enzymes ourselves. So we encode for 5 different paralogs, FMO 1, 2, 3, 4, and 5, and we have a 6th copy, which is considered a pseudogene, a bit debated, but let's say that is a pseudogene. These enzymes, along with the P450s, actually form our detoxification system, mainly in the liver, lungs, and brain, for example, and they are in charge of detoxifying different xenobiotics of toxic compounds. So basically the reaction they perform is the N-oxidation of amines to N-oxides, and also from sulfides to sulfoxides. But FMOs are actually part of a bigger class of monoxygenases, which are class B monoxygenases. All of them are FAD-dependent enzymes that use NADPH as hydrate donor, and they are related to Baeyer-Villiger monoxygenases and N-hydroxylating monoxygenases. So when we decided to go for these proteins, why was that? So basically, the reason behind it is that so far we know almost nothing of the human FMOs. And that is because these proteins have remained elusive to the characterization because it's very hard to express them and purify as recombinant proteins because these are membrane-bound proteins. So we know some things about purifications that have been done with FMOs, but we don't know too much, honestly. So our approach was that maybe we include evolutionary history into understanding this protein family, we can be able to get more information, let's say, of the biochemistry and structure of this protein. So our first thing to do was actually to explore the evolutionary history of the whole protein family. And with this, I mean, we studied how these enzymes evolve in all organisms, bacteria, archaea, and eukarya. And then we focus specifically on the animals FMOs because, of course, our interest was the human FMOs. So here you can see this is the phylogeny of the jaw vertebrates, FMOs, and you can see that In pink, we have the mammals, and here are all the parallels, FMO3, 4, 1, 2, and 5. You see clearly that FMO6 is a specific duplication occurred only in mammals from FMO3. But this is very interesting. We found that this little topology, including mammals, birds, turtles, and amphibians, is repeated for all the 5 paralogs. That means then that the common ancestor of mammals, birds, turtles, and amphibians, so the major classes of tetrapods, actually already encoded the 5 genes of FMOs. Yes? And this is the point of the emergence of the first tetrapod on Earth is around here. So this is very interesting. Something happened when animals move from the, let's say, living in the water towards the land, actually, that it caused an explosion, yes, in the number of copies of these genes into their genomes. So basically, all modern species inherit the 5 copies of these genes. And of course, the closest ancestors, which are the fishes, they have only a single gene encoding for an FMO. So that gave us a picture of, okay, these enzymes seem to be very important, and not only for mammals, but for all tetrapods there. So we started with our project with the conservative approach, and that means that we went for the ancestral sequence reconstruction for the mammalian ancestors, the common ancestor of FMO36, 4, 1, FMO2, and FMO5, which is the first diverging FMO. And we were able to reconstruct these ancestors with very good probabilities, and then we went for the expression in vitro. So we did the expression and purification, and out of the 5 ancestral FMOs, we got 4 of them correctly expressed. And this is just to show you that the process, we have to develop a whole protocol to get actually the proteins purified from the membranes, because these are membrane-bound proteins. And we did it successfully for these 4 FMOs, except for the ancestral FMO4. So the first thing we did was actually a simple thing: measure what was the thermal stability. And something came up to something really caught our attention. For ancestral FMO36, we actually have the data of the human counterpart FMO3, same for human FMO5. And we found that the ancestors, the mammalian ancestors, were actually way more thermostable, more than 10 degrees, than the modern counterparts of these proteins. You can see that here and here with the arrows. So that was a very interesting thing. But the second thing that caught our attention was that actually the proteins, when they were incubated with NADP+, actually improved their thermal stability. So by many degrees, and that happened for all the 4 ancestral proteins. Yes, more than 5 degrees. So we found out that there was a stabilizing effect of NADP+ towards thermal stability, but we also find that our ancestors were highly thermostable. And although the thermal stability cannot be related to, let's say, we cannot address the biological, we don't know if it's biologically meaningful regarding the ancestors, we knew that these conditions maybe are very good starting points to think about crystallizing these proteins. So of course, we then went for the crystallization experiments, and we actually succeeded into getting the structure of the 4 ancestors that we got. And just to show you here some peculiarities, because this was the first time that we actually knew how is the structure of an FMO, membrane bound, of course. So we found that how these proteins are sitting in the membrane, and the very interesting thing was that they are not only anchored to the membrane by the transmembrane C-terminal domain, but also they are anchored by several points in their structure. And that was very interesting to us. Also, we find that the difference among the 5 paralogs is that they display a different tunnel arrange to actually get access of the hydrophobic substrates and release the hydrophilic products. And that varies from one paralog to the other, yes? And of course, we went then for the biochemistry and biocatalysis. We performed steady-state kinetics and pre-steady-state kinetics. And basically, we saw 2 things that were very important for us. The first thing is that in general, they display the same activity and the same substrate scope, our ancestors, and the human proteins. So that's interesting because our ancestors are very good models to actually now work and understand and think about the human proteins, for example, for drug design. But also, we found that ancestral FMO1, 2, and 3, 6 actually perform the typical reactions of FMOs, which are N and S oxidation, while ancestral FMO5 performs the viability oxidation, which has been already demonstrated for human FMO5. So we were recovering these properties, and we know the ancestors are doing this. And just to show you something particular, for example, from ancestral FMO1, in ancestral FMO1, we find out that it has a broader, a bigger, more open, let's say, substrate cavity. So actually, it can accept different substrates, which are drugs like benzidamine and tamoxifen, and completely transform them very well. So we can say that from this specific example that I'm showing you today, that by incorporating ASR into our project and designing our project on an evolutionary basis, we were able to get the structure for the first time of mammalian FMOs. And actually, we understood the mechanism of access and of the substrates and product releasing. We also understood which are the biochemical differences among the 4 paralogs and how they work differently. And if we think in a more evolutionary point of view, we understand that there are 2 trajectories. One that goes from a common ancestor to FMO1 to 4, and the other one, and they all display the same kind of functionality, and the other that goes from the same ancestor to FMO5 that actually displays a different functionality. And I will come back to that a bit in a second. And also we understood how is the taxonomic distribution of FMOs among all tetrapods, because now we know that having 5 paralogs is not an exclusive feature of mammals, it's actually from all tetrapods on Earth. So with these points, we actually now are moving, we decided we were ready to go further back in time. And this is the work that we are currently doing at the moment. So as I said, we have a common ancestor that gave rise to FMO4, 1, 2, 3, and FMO5, and they have different activities. So actually, we got the ancestors, we performed the ancestral sequence reconstruction and got the ancestors that are marked as the yellow circles, and we tested them for their activity. And these are very preliminary results, but basically what we see so far is that the common ancestor of all tetrapods had a phenotype more similar to that of FMO5, to modern FMO5, than the other FMOs. So actually, of course, it's not black and white. This has a bit different properties, but actually now we are working in understanding which where the substitution occurred in that branch that led specifically to the function switching in the protein family. So, in general, some conclusions I would like to mention that as a summary, ASR is a very powerful approach. It's important to always keep in mind that the goal is to determine the phenotype rather than the precise sequence of the ancestor, and that's because it's very robust to uncertainty. And to me, I think it's very interesting that provides valuable insights to be applied in biocatalysis. For example, in understanding how new functions emerge in a protein family. And it's important to keep in mind that it's an evolutionary-based framework, so it's a historical biochemistry approach, and it's not a technique to design more stable or design in general proteins, because we're not designing anything, we are just recovering what the evolution did over time, yes? And of course, it's not exclusively computational, it requires experimental work because it's a fundamentally based on the protein function, addressing what's the protein function, yes? So that's all, and I would like, of course, to acknowledge some people, and particularly Gauthier and Callum, which were the guys involved in all the experimental work done with the FMOs, and of course Marco and Andrea Matevi, which have been involved in the FMO project, and we have been working together very nicely, and we will continue doing that. And the other people that was involved in different ASR projects from the other groups, and my colleagues in Argentina, and our funding agency. And with that, I'm finished. Thank you very much.
Okay, thanks very much, Laura, for a great talk. I'll just pass you over to Andrew, who will coordinate the questions.
Sure. Yeah. So we actually we have one question from a panelist. Anya wanted to ask a question. You can go ahead.
Sure. Thanks, Laura, for a great talk. It actually happened completely by accident to match your talk with Codex's talk, where we see an emergence of a new function in through natural evolution compared to emergence of a new function through directed evolution. So I was curious, your analysis, how quickly you could perform an analysis of, say, glucose dehydrogenase to figure out when the divergence between imine reductase or glucose dehydrogenase occurred, or and then potentially propose mutations that happened in ancestral enzymes to potentially accelerate directed evolution. Can you comment a little bit on that, please?
Yeah, thank you. Thanks. That's a very interesting question. So basically, I think that the matter of time is actually to be able to build a very nice phylogeny. So that's a very nice, I mean, very robust. You have to sample very well the data set. And once you have that, and you have cleared the questions that you want to address, it's quite fast to do ASR. But the most important thing is that you are very familiar with the protein family you are working with, because that's the only way where you actually can understand the results. For example, if you see a moonlighting activity, which would be actually, I think, the case of the GDH that is actually performing the IRED activity, I think that actually can be traced back in time and see when that emerged. Maybe it's a modern feature and it's just something that happened really, I don't know, not so far away in time, or actually developed from a different protein family. So it's very important to spend time in the constructing the phylogeny. Yes, I would say that's the most, that's the only, let's say, mostly time-consuming process during the whole approach.
Excellent, thank you.
Okay, we have another couple questions in the question and answer box. Ravi asks, what is the rationale for higher thermal stability of these enzymes with NADH+? Human FMOs aren't showing this behavior.
Well, in general, it has been more, we have seen that more often for PBMOs, for example, but during the mechanism, the releasing of NADP+ is actually the rate limiting step. So the protein has very high affinity for that. So when we inspected the structure, we couldn't see any massive difference that was telling us why that was happening compared to NADP+. But yeah, we see the stabilizing effect, and we think it's because of the affinity of NADP+, basically.
Yep. Another one from Sumitra. Are ancestral proteins necessarily more stable than their recent analogs? Why is this the case?
No, I would say no in general. And often people tend to think that ancestors are more thermostable because sometimes we recover from ASR experiments more thermostable ancestors. But that also has been proposed to be because of a bit of a bias towards the most represented residues over your phylogeny. That means that all the residues that we see nowadays in the proteins have gone through a process of evolution. So if you go backwards in time, actually you are biased a bit on the sampling because you only are comparing sequences that you know exist nowadays. So I would say that no, as a general rule, it could happen to you as it happened to us and has happened to many others. But no, it's not a technique actually or an approach to get more thermostable proteins.
Mm-hmm. Yep. Okay, another one here. Bernard asks, when you look at your different enzymes, it looks to me that the entrance of the enzymes are very different. Is this part of the protein made up of a loop structure? Do you see any indel mutants involved in all the changes?
So indels, I think that if you, I'm not sure exactly, but basically I think that she refers to this, to the access point of the substrate. And we actually see, so we see two things, that some of them share the substrate scope, but actually with different activities. So I think that's related to the substrate tunnels, as you can see here, and the releasing points, the releasing, tunnels of the products. But for FMO5, which is quite different, we expected that a bit more, and actually we were able to think about how the substrates get in and get out. But now we are working in understanding better which are the specific differences among the proteins that actually you can tell other things about how they behave biochemically. Yes. And about the length, I don't know if that was also a question, but yes, it's true that they are not all the same length, but actually that was parsed by in the ASR. And for example, for the one that we couldn't express, the FMO4, actually it has a C-terminal extension that other authors have proposed that is the reason that this protein is not expressed. And actually we truncated the protein and still is not well expressed. So it's not the reason that.
So in order to give us time for our closing remarks, I'm just going to ask one more question and then we can move on to the wrap up here. So can you do this ancestral reconstruction based on structural motifs, assuming that this is a sufficiently high number of modern enzyme structures were available?
Yes, you can do it in any protein family as long as you have the sequences, basically. You don't even need the structures. If you have it, it's better. Yes, definitely. But the approach is sequence-based.